Welcome, curious minds, to another deep dive. Today, we're not just dipping our toes in, we're plunging headfirst into one of the most complex and frankly most formidable topics in infectious diseases, the world of Clostridium bacteria, and specifically the incredibly severe infections they unleash, particularly gas gangrene.
Absolutely. It's a critical area.
Our mission, as always, is to dissect the source material, extracting every crucial nugget of knowledge, every surprising fact, so you walk away not just informed, but genuinely fluent in this intricate area. Think of it as your shortcut to becoming, well, an elite professional in understanding these microscopic adversaries.
Exactly. And this isn't just about, absorbing facts lists. It's about understanding the intricate biology of these organisms, their global reach, how they operate, and critically, what their presence means for human health and clinical management.
Right.
We'll be connecting the dots, revealing why seemingly small details are utterly crucial for both understanding these infections and, most importantly, for effectively fighting them. So, let's begin by unraveling the mysteries of this unique bacterial family.
All right, let's unpack this fascinating family, the genus Clostridium. When we talk about Clostridium, we're actually referring to an astonishingly diverse group encompassing over 60 distinct species. It's quite a range.
It really is. And what's truly striking about them is their dual nature. Some are quite benign, living quietly as part of our normal gut microflora, doing no harm at all.
Just hanging out.
Pretty much. But then there's the flip side. Many are formidable pathogens, capable of unleashing devastating, often deadly diseases.
It's like having a friendly neighbor who also happens to be a master of biological warfare.
That's actually a pretty good analogy. And that biological warfare aspect is precisely what sets the Clostridium genus apart as such a significant threat. They possess a prodigious ability to produce what the sources describe as a plethora of proteinaceous exotoxins.
A plethora sounds like a lot.
It is. That's not just a fancy way of saying a lot of toxins. It signifies a vast, diverse arsenal of protein-based presence. These aren't just a handful. This extensive array is their primary weapon, designed specifically to disrupt and destroy host tissues.
And their toxin strategies are really something else, aren't they? A masterclass in microbial virulence, as you said. Take Clostridium tetani.
Right, the cause of tetanus.
Or Clostridium botulinum, which causes botulism. These are classic examples where a single incredibly potent neurotoxins, in their case, is responsible for a very specific yet devastating clinical disease.
Exactly. Just one highly targeted toxin can completely hijack and shut down critical bodily functions. It's terrifyingly efficient.
But then in stark contrast, you have species like Clostridium perfringens and Clostridium septicum.
These are the principal culprits behind those aggressive necrotizing infections, meaning they cause rapid, widespread tissue death.
Necrotizing, yeah, that sounds bad.
It is. But their pathogenicity isn't attributed to one lone, super potent toxin like tetanus or botulism. Instead, it's a terrifying synergy, a combination of multiple toxins working in concert.
So, a team effort.
A deadly team effort. This includes bacterial proteases. Think of them as molecular scissors that break down proteins, phospholipases, which degrade the very structure of cell membranes.
The building blocks of cells.
Precisely. And various cytotoxins, which directly poison and damage cells. It's a multi-pronged, coordinated assault that just overwhelms the body's defenses.
Okay, let's zoom in on the actual bacterial cell itself, the etiologic agent, as the sources call it, and understand its anatomy and behavior. Morphologically, what are we looking at?
Well, these vegetative cells are quite pleomorphic.
Meaning they vary in shape a bit, not all identical.
Exactly. They're not all perfectly uniform. They're typically rod-shaped, though, arranged either singly or sometimes in short chains. And their ends can be rounded or sometimes pointed.
Okay. And staining, that's usually key for bacteria.
It is. And here's a critical diagnostic nuance that can trip up even experienced labs. While they stain gram-positive in their early growth stages, which is their classic appearance.
The purple stain.
Right. But they may appear gram-negative or even gram-variable later in their growth cycle, or importantly when seen directly in infected tissue specimens.
Ah, so in a real patient sample, they might look gram-negative, the pink stain.
They might. And this detail is absolutely vital for laboratory diagnosis, because a gram-negative result in a clinical sample might otherwise mislead a clinician away from a Clostridial infection if they aren't aware of this variability. It's a potential pitfall.
That's a really key point. It highlights their deceptive nature. What about movement? Can they get around?
Good question. Most strains of Clostridium are motile, yes. They achieve this using peritrichous flagella.
paratricious, meaning all over the cell surface.
Exactly. Numerous hair-like appendages distributed all over their cell surface. These flagella propel them, allowing them to spread effectively within tissues.
Any unique movers?
Yes. C. septicum has this distinct ability to swarm on solid media. It forms this spreading sort of wave-like growth pattern. It's quite characteristic.
Huh. But not all of them move.
Correct. Important non-modal species include C. perfringens, the main cause of gas gangrene, C. ramosum, and C. inoculum. And it's critical to emphasize their lack of motility in no way diminishes their pathogenicity.
Because they have those toxins.
Precisely. Their potent toxin production more than compensates for any inability to move themselves.
Okay, and then there's the whole question of oxygen. They're famous for being anaerobes, right? Hating oxygen.
That's the classic view, yes. Most species of Clostridium are obligately anaerobic. This means they absolutely require an oxygen-free environment to thrive and grow. Oxygen is toxic to them.
But I sense a but coming.
There is a but. The sources highlight that Clostridial tolerance to oxygen actually varies widely across the genus. It's more of a spectrum.
Really? Like which ones?
Well, C. septicum, which we just mentioned, and C. tertium are quite remarkable because they possess a surprising degree of aerotolerance. They can actually grow even in the presence of air.
Wow. So they're not strictly anaerobic.
They can tolerate oxygen for growth, though they won't form spores under those conditions. And this aerotolerance has profound implications for where these bacteria can survive and initiate infections in the human body. It allows them to thrive in tissues that might not be as strictly anaerobic as you typically expect for a clostridial organism.
That changes things. Makes them potentially more versatile invaders.
It does, and this adaptability brings us directly back to their unparalleled toxin arsenal. I really can't overstate this. Clostridia produce more protein toxins than any other bacterial genus known.
More than any other.
That's what the sources state. The sheer volume is astounding. Scientists have identified more than 25 distinct Clostridial toxins lethal to mice, which truly underscores the genus's potent and diverse chemical warfare capabilities.
Okay, give us a sense of what these toxins do. It sounds like a huge list.
It is an incredible laundry list of functions. We're talking about neurotoxins that attack the nervous system, like tetanus and botulinum. Enterotoxins that wreak havoc in the gut, causing diarrhea. Cytotoxins that directly destroy host cells.
Just kill them outright.
Then you have collageneses that literally break down connective tissue, the body's scaffolding. Permeases that alter the permeability of cell membranes. Necrotizing toxins that cause tissue death. Lipases and lysithinases that target fats and phospholipids in membranes.
Breaking down barriers.
Exactly. Hemolysins that explode red blood cells. Proteinases that degrade proteins.
Yeah.
Hyaluronatases that break down the glue between cells, letting them spread. DNases that chew up DNA. ADP ribosyltransferases that chemically modify and hijack host proteins. And even neuraminases that cleave specific sugar molecules on cell surfaces.
Wow. It's like a full spectrum assault kit. Each one's specialized.
Precisely. Each plays a specific role in their collective destructive capacity. And among this vast array, it's absolutely crucial to understand that botulinum and tetanus neurotoxins are the most potent toxins known to humanity, period.
The most potent known.
Yes, we're talking about extraordinarily low lethal doses for humans, maybe 0.2 to 10 nanograms per kilogram of body weight.
Nanograms. That's unbelievably tiny.
It is. To put that into practical chilling terms, a tiny, almost immeasurable speck of these toxins can be fatal. This is precisely why diseases like botulism and tetanus are so devastatingly severe and can progress with such frightening rapidity. It makes them significant public health threats, even though they're relatively rare compared to other infections.
Okay, now you mentioned something earlier, epsilon toxin. That sounded particularly concerning.
Yes, epsilon toxin. This is where it gets really interesting and quite concerning from a public health and even, frankly, a national security perspective. This is a 33 kilodalton protein produced by C. perfringens types B and D.
Not the usual type A from food poisoning?
Correct. Types B and D. Its effects in animals are just devastating severe edema, massive hemorrhage in vital organs like the brain, heart, spinal cord, kidneys.
Total system damage.
Pretty much. The sources highlight its classification as among the most lethal of the clostridial toxins. And here's the chilling part. It's explicitly considered a potential agent of bioterrorism.
Bioterrorism, wow.
Yes. This truly elevates C. perfringens, or at least these specific types, from a common food bug or wound contaminant to a microorganism with significant public health and national security implications.
That definitely raises the stakes.
Absolutely. And to fully grasp their virulence, all these mechanisms, it's worth noting that genomic studies are proving to be crucial. Looking at their DNA.
Right, sequencing their genes.
Exactly. These studies allow for a comprehensive deep dive into the complex array of virulence factors involved in Clostridial pathogenesis. It reflects the ongoing cutting-edge scientific effort to unravel their deadly secrets and ultimately find new ways to counter them.
So, okay, we know they're dangerous. Where do these notorious organisms actually lurk? Let's dive into their epidemiology and transmission, because understanding their habitat and how they move around is key to understanding how they cause disease.
Definitely. First off, Clostridium species are incredibly widespread in nature. Ubiquitous, really, everywhere, pretty much.
Yeah.
Their ubiquity is primarily due to their remarkable ability to form those resilient endospores we mentioned.
Right, the survival capsules.
Think of them as tiny, virtually indestructible survival capsules that allow the bacteria to persist in the harshest environments. Heat, drying, chemicals for extended periods, sometimes years or even decades.
Wow, so where do we find them?
You'll commonly find these spores almost everywhere. In soil, animal feces, sewage, marine sediments. They're literally a part of the fabric of our natural world.
So, inescapable, basically.
To a large extent, yes. And the prevalence of these spores, and consequently the incidence of the diseases they cause, is heavily influenced by environmental factors.
Like what?
Take C. perfringens again. Its ecology in soil is directly influenced by the degree and duration of animal husbandry in a given area. More animals, more manure, potentially more spores in the soil.
Okay, makes sense.
And this directly correlates with the incidence of gas gangrene. For example, the incidence of Clostridial gas gangrene is notably higher in the agricultural regions of Europe, maybe linked to contamination of war wounds back in the day, compared to a place like the Sahara Desert, where soil conditions and animal activity are very different.
So the environment directly shapes the risk.
Absolutely. Similarly, the incidence of tetanus and foodborne botulism are clearly linked to the widespread presence of Clostridial spores in soil, water, and various foods we consume daily. Improperly canned food, for instance, is a classic source for botulism spores germinating.
Now, here's a crucial point you touched on that might surprise many. Clostridia aren't just out there in the dirt. They're also a very common part of our own internal landscape, right? Our normal microbiome.
That's exactly right. They are present, often in large numbers, in the indigenous microbiota of the human and animal intestinal tract. They're also found in the female genital tract, and even on the oral mucosa.
In our mouth.
Yes, they literally live inside and on us, often without causing any trouble at all. They're just part of the complex ecosystem.
Which leads to that critical distinction you made earlier.
Yes, and it cannot be overstated.
Yeah.
Not all commensal Clostridia, meaning those that live harmlessly with us, are toxigenic. They don't all make the dangerous toxins.
So just having them isn't the problem.
Correct. Just because you have Clostridium species in your gut or elsewhere doesn't automatically mean you're going to get sick. It's only when specific toxigenic strains, often under very particular predisposing conditions like tissue damage, creating an anaerobic environment, or an altered immune state, or antibiotic use disrupting the normal gut flora proliferate, or are introduced into a vulnerable site, that they become dangerous.
Okay.
This distinction is absolutely key for understanding when harmless colonization transitions into a dangerous infection. It's all about context and the specific strain.
So what does this widespread presence, both outside and inside us, mean for public health on a global scale? Are these still major problems?
They remain a serious global public health concern, absolutely. In developing nations, the problem is particularly pronounced. Conditions like food poisoning, necrotizing enterocolitis in infants, and gas gangrene are distressingly common.
Why is it worse there?
Well, factors like poverty, inadequate sanitation, lack of clean water, and limited access to healthcare, including things like prompt wound care or safe food handling practices, create ideal conditions for their prevalence and severity. The challenges in these regions are truly systemic and deeply embedded.
But it's not just a problem in developing countries, right? The sources mentioned they're still prevalent in developed nations too.
That's absolutely right. These aren't just problems confined to the developing world. These infections remain prevalent even in highly developed countries. Gas gangrene, for instance, commonly follows traumatic injuries. Think knife or gunshot wounds, serious vehicular accidents.
Major trauma.
Yes. Or it can tragically develop as a complication of surgery, especially abdominal surgery, or even arise from an underlying gastrointestinal carcinoma that might provide a portal of entry.
And are there new threats emerging?
Yes, we're seeing emerging threats too. Severe Clostridial infections have become a significant health threat to specific vulnerable populations.
Hmm.
We already mentioned injection drug users.
Right, introducing bacteria directly into tissues?
Exactly.
Yeah.
Especially with things like black tar heroin injected subcutaneously, skin popping.
Hmm.
And also women undergoing childbirth or abortion, particularly if proper sterile techniques are not rigorously maintained, or if significant tissue damage occurs, creating that anaerobic environment.
We also need to consider the historical context, don't we? Gas gangrene and war.
Oh, absolutely. Historically, Clostridial gas gangrene has been notoriously known as the scourge of the battlefield. It devastated soldiers in World War I and World War II. It was a terrifying reality for battlefield medics before modern wound care and antibiotics became widespread.
And the sources suggest this isn't just history.
No, looking to the future. The sources directly connect this to modern global political situations. They explicitly postulate a possible scenario involving mass casualties from war or terrorism that could lead to extensive, contaminated injuries highly conducive to gas gangrene.
So, a potential future threat as well.
It's a very real, ongoing, and terrifying threat in today's world, unfortunately, not just a historical footnote.
Given all this, their ubiquity, their potency, the ongoing threat, what can be done, is prevention possible?
It's a major focus. Given this persistent and evolving threat, there is an ongoing and critical need to develop novel strategies to prevent, or at least attenuate, meaning lessen the severity of the course of Clostridial infections in both civilian and military personnel.
Like what? Vaccines.
Vaccination is one of the most promising avenues, yes. Vaccination against the specific exotoxins important in pathogenesis, like the alpha toxin in C. perfringens or the beta toxin in C. perfringens type C, would offer immense benefit.
Especially where these diseases are common.
Particularly in developing nations where these diseases are rampant, yes. But it also holds potential for at-risk populations in developed countries. Think about the elderly, patients with diabetes who might require lower limb surgery due to trauma or poor circulation.
Being potential entry points.
Exactly. Or those undergoing intestinal surgery, where the gut microbiome itself is a potential source if there's leakage.
Okay, vaccines are one approach. Anything else?
Beyond vaccines, the sources also highlight the significant value of something called a hyperimmune globulin.
What's that?
It's essentially a concentrated dose of preformed antibodies, specifically designed to target and neutralize the Clostridial toxins.
Like an antidote.
Sort of, yes. A passive immunity boost. Such a tool would be incredibly valuable for prophylaxis, essentially, preventative treatment in victims of acute traumatic injury, given before infection can even take hold.
Or if someone already has it.
Or, crucially, it could be used for attenuating the spread of infection in patients who already have established gas gangrene, helping to neutralize the toxins causing the damage. It offers a glimpse into advanced therapeutic approaches that could truly make a life-saving difference, especially given the speed of these infections.
Okay, let's transition now to the specific clinical syndromes and infections caused by Clostridia. You mentioned a whole spectrum.
Yes, it's a wide and often horrifying spectrum of clinical syndromes. These are truly life-threatening manifestations, ranging from relatively common intoxications like food poisoning,
Which most people think of with Clostridium perfringens.
right, but also tetanus and botulism, which are pure intoxications. Then you move into severe invasive infections like necrotizing enteritis and colitis bowel destruction bacteraemia, where bacteria get into the bloodstream.
A very bad sign.
A very bad sign. Myonecrosis, which is the muscle destruction we call gas gangrene, and even systemic responses like toxic shock syndrome, TSS. It's quite a range.
And just to clarify, we're focusing here on the ones in Chapter 154, right? So not tetanus or botulism specifically today.
Correct. Tetanus and botulism are covered in other chapters. Our deep dive here focuses specifically on the conditions detailed in Chapter 154 of our source material, primarily C. perfringens, C. septicum, C. sordelii, and related infections.
Got it. So let's start with a very common one many people might have encountered or heard about. C. perfringens type A food poisoning. How common is this, really?
It's highlighted as one of the most common bacterial causes of foodborne illness in the United States and Canada. So it's definitely not a rare occurrence. It's a frequent culprit behind outbreaks of stomach flu that aren't actually the flu virus at all.
And what kinds of foods are usually involved?
The typical culprits are improperly cooked or stored meat and meat products, especially things like large roasts, stews, or gravy.
Why those specifically?
The mechanism is quite fascinating. Residual spores of C. perfringens type A, which can survive cooking, germinate, and then rapidly proliferate if the food is cooled too slowly, say, left out on the counter for too long or reheated insufficiently. They love that warm anaerobic environment inside a thick stew or roast.
Okay, so the spores survive, then the bacteria grow in the food.
Exactly. Illness then results from ingesting a large number of these viable vegetative cells. The source says at least around 100 million, or 108 cells.
That's a lot of bacteria.
It is. Once ingested, these cells encounter the alkaline environment of the small intestine. This triggers them to sporulate, form new spores, and it's during the sporulation process in the gut that they release the C. perfringens enterotoxin, CPE.
So it's toxin production in the gut, not preformed toxin in the food like some other food poisonings.
Precisely. It's the toxin released during sporulation in the small intestine that directly causes the clinical symptoms.
And what are those symptoms typically like?
The clinical course is usually quite mild and self-limiting. Typically presents as watery diarrhea, abdominal cramps. Nausea is less common. Vomiting and fever are rare. It usually develops within 7 to 30 hours, maybe 8 to 22 hours, is most typical after ingesting the contaminated food.
Sounds unpleasant, but manageable for most people.
For most healthy adults, yes. It's usually an uncomfortable day or two. However, and this is a critical point, often overlooked symptoms can be much more severe and occasionally even fatal in particularly vulnerable populations.
What are we talking about?
The very young, the elderly, and individuals who are immunocompromised. For these groups, the dehydration and stress from the illness can be much more dangerous, so it's not always benign.
Any chronic issues linked to it?
Interestingly, yes. It's also implicated as an etiologic agent of persistent diarrhea in elderly patients, particularly those in nursing homes and tertiary care institutions. And it's even been considered to play a role in some cases of antibiotic-associated diarrhea, even when the more famous culprit, C. difficile, isn't found, or when pseudomembranous colitis isn't present.
So it can be a factor even outside of typical food outbreaks.
It seems so. Delving into the molecular basis, those strains of C. perfringens associated with food poisoning possess a specific gene, known as CRAPE, which codes for that enterotoxin.
The CPE toxin.
Yes. And this enterotoxin acts by binding to receptors on intestinal cells and forming pores in the cell membranes, disrupting ion transport and leading directly to fluid loss and diarrhea.
And you mentioned something about plasmids earlier.
Right. What's also intriguing and complicates control efforts is that C. perfringens strains isolated from non-foodborne diseases, like those cases of antibiotic-associated or sporadic diarrhea, often carry this CRAPE gene on a plasmid.
A mobile piece of DNA.
Exactly. A small circular piece of DNA that can be easily transferred between different bacteria through horizontal gene transfer. This plasmid-mediated transmission means the ability to cause this type of diarrhea can potentially spread to other C. perfringens strains, making control harder.
How do labs detect it then if someone has persistent diarrhea, for example?
For laboratory detection of the toxin in feces, various methods are used. There are cell culture assays using vero cells, enzyme-linked immunosorbent assays, ELISA, reverse-phase latex agglutination tests, and also PCR amplification to detect the CRAPE gene itself directly in stool samples. Each method has its own advantages and limitations in terms of speed, sensitivity, specificity, and cost.
Okay. Moving on to something far more devastating. Enteritis necroticans. The source has also called this gas gangrene of the bowel. That sounds truly awful.
Yes, this is a fulminating clinical illness characterized by extensive necrosis, meaning widespread tissue death of the intestinal mucosa and the intestinal wall itself. It's truly a gut-destroying infection that develops with terrifying speed.
How does it typically occur?
Cases can occur sporadically in adults, or horrifyingly as epidemics affecting people of all ages, especially in certain populations or settings.
And what causes this specifically? Is it C. perfringens again?
Yes, but a different type and different toxins are key here. The etiology for this severe condition is specifically attributed to alpha toxin and beta toxin producing strains of C. perfringens type C.
Type C this time. And beta toxin is the main culprit.
Crucially, yes. The beta toxin, like the CRAPE gene in some strains, is also plasmid located, and it is considered primarily responsible for the pathogenesis of enteritis necroticans. It causes ischemic necrosis tissue death due to lack of blood flow, primarily in the jejunum, which is a part of the small intestine.
Is this the disease called pig bell?
Exactly. Historically, this condition had a devastating impact in Papua New Guinea, particularly during the 1960s, where it was known locally as pig bell.
Why that name?
It was strongly associated with traditional pig feasts, where large amounts of pork, sometimes undercooked, were consumed. It became tragically the most common cause of death in childhood there during that period, occurring both sporadically and in sudden, deadly outbreaks linked to these feasts.
That's heartbreaking. Was anything done about it?
Yes, and here's a genuine public health success story mentioned in the sources. Intramuscular immunization using a toxoid vaccine against the beta toxin was introduced in Papua New Guinea.
A vaccine against the toxin.
Precisely. And it resulted in a dramatic decrease in the incidence of pig bell. This really highlights the potential of targeted vaccination against specific Clostridial toxins, proving that understanding the enemy's weapons can lead to effective defenses.
That's fantastic news. But is it only found there, or just historically?
Despite that success, enteritis necroticans isn't just a historical or geographically confined disease. It's also been recognized, albeit less commonly, in developed nations like the United States, the United Kingdom, and Germany, where it is known by its German name, Darmbrand.
And are certain people more at risk, even in developed countries?
Yes, specific adult populations seem particularly susceptible. Those who are malnourished or who have underlying conditions like diabetes, alcoholic liver disease, or neutropenia, that dangerously low white blood cell count we keep mentioning. It shows the crucial role of host health and underlying conditions in susceptibility.
Okay. Now, closely related but distinct is necrotizing enterocolitis, or NEC. How does this compare?
NEC shares significant similarities in pathology with enteritis necroticans, but it has a specific association with C. perfringens type A, the same type usually involved in food poisoning, rather than type C.
Type A again, and who does it affect?
Its primary affected population is our smallest and most vulnerable patients. Low birth weight, premature infants hospitalized in neonatal intensive care units and ICUs.
Oh, newborns, that's terrible.
It's a devastating disease in these incredibly fragile patients. And frustratingly, the precise etiology and pathogenesis of NEC have actually remained quite enigmatic, quite mysterious for over 4 decades, indicating its immense complexity.
What are the similarities and differences in pathology compared to enteritis necroticans?
Both NEC and enteritis necroticans share similarities in the pattern of small bowel necrosis, affecting the submucosa, mucosa, and muscularis layers. They both also feature gas dissecting tissue planes, gas getting into the bowel wall, and a similar degree of inflammation.
But there are contrasts.
Yes, crucial contrasts. Unlike enteritis necroticans, which most commonly involves the jejunum, NEC frequently affects the ileum and often the ileothecal valve, the junction between the small and large intestines. Also, intestinal gas cysts, called pneumatosis intestinalis, those small pockets of gas within the bowel wall are generally more common and prominent in NEC.
Where does that gas come from?
The gas itself, which typically contains hydrogen, methane, and carbon dioxide, is likely a byproduct of the fermentative activities of various intestinal bacteria. This includes Clostridium, not just C. perfringens, but also C. neonatal, and certain other Clostridia have been implicated, or even other bacteria like Klebsiella species. It's likely a complex interplay.
How is NEC managed in these tiny infants?
For suspected Clostridial enteric infection, like NEC, the recommended treatment approach is multifaceted and supportive. It involves nasogastric suction to decompress the bowel, intravenous fluids to support hydration and circulation, sometimes oral antibiotics like parenteral, and critically, bowel rest, achieved by stopping feeds and providing nutrition intravenously, parenteral nutrition. Intravenous antibiotics, often including agents active against Clostridia like benzylpenicillin or others depending on local patterns, are administered.
And close monitoring is key.
Absolutely critical. The infant is closely observed for any complications that might necessitate surgery, things like bowel perforation. Thankfully, infants with milder cases can recover completely without surgical intervention.
And if surgery is needed.
However, if surgical indications are present, such as free air detected in the peritoneal cavity indicating a perforation, absent bowel sounds, rebound tenderness, or abdominal rigidity, the mortality rate is tragically high, ranging from a devastating 35% to as high as 100% in some series, often due to the complications of intestinal perforation and subsequent sepsis. This really underscores the critical, life-saving importance of rapid diagnosis and decisive intervention in this fragile population.
It sounds incredibly challenging. Let's shift gears slightly. What about infections where Clostridia are involved, but maybe not as the sole actor? Polymicrobial infections?
Yes, that's a very common scenario. It's important to understand that Clostridial species often appear in association with other bacteria specifically, non-spore-forming anaerobes like bacteroids or Prevotella, and facultative or aerobic organisms like E. coli or Staphylococcus in these complex mixed infections.
So they're part of a team, so to speak.
Often, yes. They are rarely the sole culprits in these specific scenarios. They're frequently part of a formidable, mixed microbial team, with each member potentially contributing to the overall pathology through synergistic interactions.
Where do you see these mixed infections happening?
These Clostridia may be involved in a remarkably diverse array of infection sites. Everything from head and neck infections like deep neck space abscesses to conjunctivitis in the eye, brain abscesses, sinusitis, otitis media.
Lungs too.
Yes, aspiration pneumonia, lung abscesses, pleural empyema pus collecting around the lung, also cholecystitis, gallbladder infection, septic arthritis in joints, and various bone infections like osteomyelitis.
Do these mixed infections look different clinically?
That's a key point. What's particularly nuanced clinically is that these mixed conditions are often associated with severe local inflammation, but they may surprisingly lack the characteristic systemic signs of profound toxicity and rapid progression that we see in classic clostridial infections like gas gangrene.
So maybe less shock, less overwhelming sepsis initially.
Often, yes. The local infection can be severe, but the systemic picture might be less dramatic, at least early on. This subtle difference is a key diagnostic challenge. You might underestimate the clostridial component if you're only looking for the classic signs.
That's crucial for clinicians to remember. Where else are these found? Abdomen.
Very commonly. Clostridia are isolated from approximately 2/3, 66%, of intra-abdominal infections, particularly where the mucosal integrity of the bowel or sometimes the respiratory system has been compromised. Think after surgery, trauma, or perforation like diverticulitis.
Which species are most common there?
In this setting, C. ramosum, C. perfringens, and C. bacteraemia are the most commonly isolated species. But again, it's noted that their mere presence in these mixed infections doesn't invariably lead to a poor outcome. It truly depends on the specific microbial synergy, the overall bacterial load, and various host factors at play. It emphasizes the polymicrobial nature and the variable severity.
Any other common sites for these mixed infections?
They've also been isolated quite frequently from suppurative infections, meaning infections producing pus of the female genital tract, for example, ovarian or pelvic abscesses, tubo-ovarian abscesses, and also from diseased gallbladders.
You mentioned cholecystitis earlier.
Yes. While C. perfringens is the most frequently isolated species in gallbladder infections, full-blown gas gangrene of the gallbladder isn't typically observed there. However, the presence of gas-forming Clostridia in the biliary system can lead to a condition known as emphysematous cholecystitis.
Gas in the gallbladder wall.
Exactly. Gas accumulates in the gallbladder wall, visible on imaging. This is especially common in diabetic patients, likely due to vascular compromise and altered metabolism favoring anaerobic growth.
And what about really aggressive soft tissue infections?
Yeah.
Necrotizing fasciitis?
Absolutely. C. perfringens, often in association with mixed aerobic and anaerobic microbes, can cause aggressive, life-threatening tight ren. Necrotizing fasciitis, that's the polymicrobial type, or Fournier's gangrene, which is necrotizing fasciitis of the perineum and genital area. These are rapidly spreading and devastating soft tissue infections requiring immediate surgical intervention.
So, when faced with these suspected mixed infections, abdomen, perineum, gynecologic organs, how should treatment be approached, especially before you have culture results?
Empirical treatment guidelines are crucial here. The approach must be strategic and broad enough to cover the likely players. It should ideally be based on initial clues like grams, staining of exudate, the clinical site, and known risk factors, followed by tailoring based on culture results and antibiotic sensitivity information when they become available.
What's typically recommended to start with?
The recommended empirical therapy, as outlined in Table 154-1 from the source, typically consists of an agent with good gram-positive and anaerobic coverage, like ampicillin or ampicillin sulbactam, combined with an agent specifically targeting anaerobes, like either clindamycin or metronidazole. This provides broad coverage for both the aerobic and anaerobic components expected in these often complex infections.
What if you need even broader coverage? Maybe for resistant gram-negatives?
Right. Broader gram-negative coverage might become necessary if the patient has specific risk factors, like recent hospitalization, prior antibiotic treatment, or known colonization with resistant organisms, suggesting a more complex or resistant flora.
How do you achieve that?
This can be achieved by substituting broader spectrum agents like T-carcillin clavulanic acid, piperacillin tazobactam, often preferred now over tecarcillin clavulanate, or a carbapenem antibiotic like imipenem or meropenem for the initial ampicillin component. or alternatively by adding a fluoroquinolone or an aminoglycoside to the existing regimen, though aminoglycosides have limitations in anaerobic environments like abscesses.
And how long is treatment usually needed?
Empirical treatment is generally continued for about 10 to 14 days, or often guided by the patient's clinical response continued until the patient's clinical condition significantly improves, showing clear resolution of the infection signs and symptoms.
Okay, next up, Clostridial bacteremia. When these bugs actually manage to get into the bloodstream, how significant is that?
It's a very significant concern, yes. Clostridium species are important causes of bloodstream infections, and their presence in the blood often signals a deeper, more severe underlying problem, or is a precursor to widespread dissemination.
Which ones are most common in blood cultures?
Molecular epidemiologic studies have identified C. perfringens, which accounts for a large proportion, up to 79% in some studies, of Clostridial bacteremias, and C. tertium, around 5%, as the two most frequently isolated species in these critical cases.
What's the prognosis if C. perfringens is in the blood?
Well, C. perfringens bacteremia can occasionally occur without an immediately identifiable infection site, maybe transiently from the gut. But when it's associated with myonecrosis, the severe muscle death of gas gangrene, it carries a very grave prognosis. It means the infection is systemic and overwhelming.
You mentioned C. septicum earlier as being particularly concerning. What about its presence in the blood?
Yes, C. septicum bacteremia is a particularly crucial diagnostic red flag for clinicians. This species is rarely isolated from the feces of healthy individuals, although it might be found incidentally in the normal appendix sometimes.
So finding it in blood is unusual.
Very unusual and highly significant. What's striking is that over 50% of patients with positive blood cultures for C. septicum have some underlying gastrointestinal anomaly. This could be diverticular disease, inflammatory bowel disease, or, more ominously, an underlying malignancy, most notably carcinoma of the colon or other GI cancers.
So C. septicum in the blood means you have to look for cancer or a gut problem.
Absolutely. It should immediately trigger an aggressive search for these underlying conditions, as the bacteremia is often the first sign of that hidden pathology.
Any other conditions linked to C. septicum bacteremia?
Yes, another clinically important association is with neutropenia of any origin, that low neutrophil count again. More specifically, it's associated with neutropenic enterocolitis, also called typhlitis, an inflammatory condition of the bowel, often involving the terminal ileum or cecum, especially in patients undergoing chemotherapy.
So immunocompromised patients are at high risk.
Very much so. Other significant risk factors include patients with diabetes mellitus, severe atherosclerotic cardiovascular disease, or existing anaerobic myonecrosis, gas gangrene. It has also been recovered from the bloodstream of patients with liver cirrhosis, alongside C. perfringens, C. bifurmentans, and other Clostridia.
And didn't you link some Clostridial bacteremia to toxic shock syndrome?
Yes, specifically bloodstream infection by C. sordelii and sometimes C. perfringens has been directly associated with toxic shock syndrome, a life-threatening systemic inflammatory response.
Now, what about C. tertium bacteremia? You said it was challenging.
Ah, yes. C. tertium presents a particularly challenging exception within Clostridial bacteremia. It can be found in patients with serious underlying diseases like malignancy or acute pancreatitis, often, again, with or without neutropenic enterocolitis. However, its exact frequency hasn't been systematically studied, making it harder to pin down its true prevalence.
What makes it so difficult?
The real problem with C. tertium lies in both its identification and, critically, its treatment. This organism may deceptively stain gram-negative or gram-variable.
Like we discussed earlier.
Exactly. It's also aerotolerant, which can confuse labs looking for strict anaerobes. And most crucially, it is intrinsically resistant to many commonly used antibiotics, including metronidazole, clindamycin, and cephalosporins.
So the standard treatments might not work.
They often won't.
Yeah.
This makes empirical treatment incredibly difficult and highlights the dire need for specific and accurate identification and susceptibility testing for C. tertium.
Are there other Clostridia found in blood?
Yes, beyond these main ones. Other Clostridia from the C. clostridioform group, which includes C. clostridioform itself, along with C. hathaway and C. boltei, are also capable of causing bacteremia, often originating from the gut as well.
So the bottom line here is
the clinical importance of recognizing any Clostridial bacteremia, but especially that due to the red flag organism C. septicum and starting appropriate treatment immediately, cannot be overstated. This is truly a life or death situation where every hour counts.
But diagnosis is still mainly by culture.
It's critical to note the diagnostic limitations here. Alternative methods to rapidly identify bacteraemia causing Clostridial species, like specific PCR assays or other rapid diagnostic tests, are not currently widely available or routinely used for all these species in most clinical labs.
So we still rely on the older methods.
This means that traditional anaerobic blood cultures combined with careful gram stain interpretation by an experienced microbiologist remain the best diagnostic tests at this point for accurate and timely identification, despite their inherent time requirements for growth.
Okay. Let's turn our attention now to the skin and soft tissues. Clostridial infections there sound particularly nasty.
They are. These are characterized by incredibly rapid destruction. The key histotoxic Clostridial species involved here, meaning those that destroy tissues, include C. perfringens, C. histolyticum, C. septicum, C. novii, and C. sordelii.
That's quite a list of bad actors.
It is. These species cause aggressive necrotizing infections, and their aggression is attributable in large part to that elaboration of bacterial proteases, phospholipases, and cytotoxins. We discussed the synergistic toxin arsenal working together to dissolve and destroy tissues.
What are the hallmarks you look for?
The hallmarks of these infections are their terrifyingly rapid progression, marked tissue destruction, the unmistakable presence of gas in the tissues, which you can often feel as crepitus.
That crackling sensation.
Exactly, and the development of systemic shock, frequently ending in death if not treated aggressively and immediately.
What are the clinical signs on the patient?
Clinically, you'll typically see severe pain, often out of proportion to the visible injury. That crepitus, the crackling from gas. Brawny induration, meaning the skin becomes hard, woody, and thickened. This rapidly progresses to skin sloughing, where layers of skin detach, and the formation of violaceous or bluish-purple bulor, large fluid-filled blisters. Patients will also often present with marked tachycardia, a very rapid heart rate, even if they don't have a high fever initially.
So pain, swelling, skin changes, gas, fast heart rate.
Those are absolutely critical signs for early recognition, as time is undeniably of the essence in these infections.
All right, now for the main event of our deep dive. Clostridial myonecrosis, more commonly and terrifyingly known as gas gangrene. This sounds like the absolute worst-case scenario.
It is truly a devastating infection and a true surgical emergency. C. perfringens myonecrosis is characterized as one of the most fulminant, meaning incredibly rapid and severe, gram-positive bacterial infections in humans.
And treatment is tough.
And here's the stark reality. Even with appropriate antibiotic therapy and intensive care support, the tissue destruction often progresses at an alarming rate. The toxins just keep working.
What are the systemic effects? Does it stay local?
No, it rarely stays local. Gas gangrene is almost invariably accompanied by bacteraemia, the bacteria entering the bloodstream. This leads to dangerously low blood pressure or hypotension, and often multi-organ failure. Kidneys, lungs, liver start shutting down.
So it's fatal if not treated.
It is, almost without exception, fatal if left untreated or if treatment is delayed. This is not a condition where you can afford to wait and see. It is a true medical emergency that demands immediate and aggressive surgical debridement.
You mentioned two key conditions needed for it to develop. What were they again?
Right. The development of gas gangrene absolutely requires two key conditions. First, an anaerobic environment, that is, a profound lack of oxygen in the tissue. And second, contamination of a wound with Clostridial spores or vegetative organisms.
What creates that anaerobic environment?
Several factors contribute. Devitalized or dead tissue from the initial injury has no blood supply, so no oxygen. Foreign bodies lodged in the wound like dirt, clothing fragments, shrapnel can impede blood flow and harbor bacteria. And ischemia itself, meaning reduced blood supply from damaged arteries, all work together to significantly lower local oxygen levels, creating the perfect breeding ground for the outgrowth of vegetative cells from any spores present.
So what types of injuries are most likely to lead to traumatic gas gangrene?
We're talking about severe crush-type injuries, where tissue is massively damaged. Lacerations of large or medium-sized arteries that cut off blood flow downstream. And open fractures of long bones, especially if they're heavily contaminated with soil or bits of clothing containing bacterial spores.
Soil is a big source of spores, right?
A major reservoir, yes. Also, gas gangrene of the abdominal wall and flanks often follows penetrating injuries, like deep knife or gunshot wounds, that are severe enough to compromise intestinal integrity, nicking the bowel. This leads to the leakage of bowel contents, which are teeming with Clostridia directly into the surrounding soft tissues.
What about non-traumatic sources or surgeries?
Proximity to fecal sources of bacteria is a significant risk factor. We sadly see cases following hip surgery, particularly if there were complications or contamination. Even things like improperly administered adrenaline injections into the buttocks have been reported, presumably introducing bacteria deep into tissue. And amputation of the leg for ischemic vascular disease, the tissue is already compromised, providing a perfect entry point if contaminating.
And you mentioned injection drug use earlier.
Yes, that's a more recent and concerning emerging risk factor. In the last couple of decades, cutaneous gas gangrene caused by C. perfringens, C. novae, and C. sordelli has been described, particularly in the U.S. and Northern Europe, specifically among persons injecting black tar heroin subcutaneously skin popping.
Why that specifically?
Black tar heroin is often contaminated, and injecting it under the skin rather than into a vein creates a pocket of damaged, poorly oxygenated tissue where the Clostridia can thrive. It highlights how new routes of exposure can lead to these classic severe infections.
The speed sounds incredible. How fast does it develop?
The rapid incubation and progression of traumatic gas gangrene are truly alarming, almost beyond belief sometimes. The incubation period can be incredibly short, as little as 6 hours after injury in some cases, though it's usually less than 4 days.
And the first sign is pain.
Often, yes. It's characterized by the sudden onset of excruciating, often disproportionate pain at the affected site. Pain far worse than you'd expect from the injury itself.
What does the wound look like?
The wound itself rapidly develops a foul, often described as sweetish or mousy odor. There's usually a thin, watery, serosanguineous sort of brownish, blood-tinged discharge. And crucially, you often see or feel gas bubbles, which produce that characteristic crackling sensation, crepitus, when you press on the skin.
And the surrounding tissue.
As the infection progresses, bronchoedema, that hard, non-pitting swelling and induration, a hardened, woody consistency develop around the wound. This quickly gives way to the formation of cutaneous blisters, or bullae, containing bluish to maroon-colored fluid. These tissues may later liquefy and slough off, literally falling away.
How fast does it spread? You said inches per hour.
What's truly terrifying is the speed of spread. The margin between the visibly healthy tissue and the advancing necrotic tissue often advances several inches per hour, even despite appropriate antibiotic therapy being administered. The toxins just keep marching forward.
Which is why surgery is so critical.
Exactly. This relentless progression is why radical surgical intervention, often requiring amputation of an affected limb, frequently remains the single best, and indeed sometimes the only life-saving intervention. You have to get ahead of the necrosis.
And the systemic effects again.
Systemic shock and multi-organ failure frequently accompany the local destruction in gas gangrene. And when patients become bacteremic, the bacteria flooding their bloodstream, the mortality rate skyrockets to exceed 50%, even with modern intensive care.
How is traumatic gas gangrene usually diagnosed then? It sounds fairly obvious if you know what to look for.
Diagnosing the traumatic form is typically not difficult for an experienced clinician, primarily because the infection consistently begins at the site of significant trauma, is associated with demonstrable gas in the tissue, clinically or on imaging, and progresses with that terrifying rapidity.
And the gram stain.
Gram's staining of drainage or a tissue biopsy is usually definitive. It classically demonstrates large, boxcar-shaped, gram-positive or sometimes gram-variable rods. And crucially, you'll observe that striking absence or paucity of inflammatory cells neutrophils right in the infected tissue itself.
The paradox you mentioned.
Exactly. It's paradoxical, given the severity of the infection, and it's a key diagnostic clue driven by the toxins, which effectively kill or repel the immune cells trying to respond. This is seen in conjunction with widespread muscle and soft tissue necrosis.
Okay, that covers traumatic gas gangrene, but you also mentioned spontaneous or non-traumatic gas gangrene. How does that happen?
Yes, spontaneous gas gangrene is an even more insidious and often tragically missed threat. Unlike traumatic cases where bacteria enter a wound, this generally occurs via hematogenous seeding.
Bacteria spreading through the blood.
Exactly. Bacteria, usually originating from the gastrointestinal tract, travel through the bloodstream and seed seemingly normal muscle tissue, where they then manage to proliferate.
Which Clostridium species do this?
The principal culprits here are C. perfringens, again, but very importantly C. septicum and also C. novae. Occasionally C. tertium has been implicated too.
Where in the gut do they come from?
The portal of entry for these blood-borne bacteria is typically from some disruption or pathology within the GI tract. Common sources include an underlying colonic malignancy, inflammatory bowel disease like Crohn's or ulcerative colitis, diverticulitis, necrotizing enterocolitis, cystitis, inflammation of the cecum, or distal ileitis, or sometimes following bowel surgery if there's a minor leak.
And why is C. septicum particularly dangerous here?
Because, as we discussed, C. septicum is aerotolerant. It can survive and proliferate even in the presence of some oxygen. This means it can potentially infect and spread through normal, healthy tissues, unlike other Clostridia that require a strictly oxygen-free environment created by trauma.
So it doesn't need that big injury to get started.
Correct. This makes it uniquely insidious because it doesn't need a pre-existing crush injury or massive tissue damage. A small breach in the gut lining might be enough for it to enter the bloodstream and then set up shop in muscle tissue somewhere else.
What does that mean for patients who survive it?
It means that patients surviving bacteraemia or spontaneous gangrene due to C. septicum should absolutely undergo aggressive diagnostic studies like colonoscopy, CT scans, to rule out any underlying gastrointestinal pathology, especially colon cancer, as this is a common and often hidden source for this particularly virulent species. Finding C. septicum gangrene is a strong warning sign for underlying gut disease.
Are there other risk factors for spontaneous gangrene besides gut problems?
Yes, additional predisposing host factors include conditions that compromise the immune system more broadly. Things like leukemia, lymphoproliferative disorders like lymphoma, patients undergoing cancer chemotherapy or radiation therapy, and individuals with AIDS.
And neutropenia, I guess.
And especially neutropenia. There's a strong and clinically important association of cyclic, congenital, or acquired neutropenia with an increased incidence of spontaneous gas gangrene due to C. septicum. In such cases, associated necrotizing enterocolitis, cystitis, or distal ileitis is common, particularly among children with neutropenia.
How does spontaneous gangrene present? Is it obvious?
That's the danger. The onset can be quite subtle, but the deterioration is frighteningly rapid. The very first symptom may be something vague, like general confusion or malaise. This is then often followed by the abrupt onset of excruciating pain, typically in a limb or the trunk, but crucially, in the absence of any trauma or obvious wound. Fever might be present, but not always initially.
Pain without injury. That should be a red flag.
It should be a massive red flag for spontaneous gas gangrene. However, because there's often no obvious portal of entry, the correct diagnosis is frequently delayed or tragically completely missed. Clinicians might think of muscle strain, a blood clot, other things first.
And that delay is critical.
Catastrophic, given the speed. The infection is characterized by rapid progression of tissue destruction with demonstrable gas in the tissue, often visibly confirmed on imaging like X-rays or CT scans. Figure 154-2 in our source shows a classic example.
What happens to the tissue visually?
Swelling increases rapidly. Those bully large blisters filled with clear, cloudy, hemorrhagic, or purplish fluid begin to appear. The surrounding skin takes on a dusky or purple hue, which reflects severe vascular compromise resulting from the diffusion of bacterial toxins into surrounding tissues, shutting down blood flow.
And it spreads quickly?
Invasion of adjacent healthy tissue rapidly ensues, with quick progression to systemic shock and multi-organ failure, just like in the traumatic form.
What are the survival rates like?
The mortality rates for spontaneous gas gangrene are extremely high. The source's site ranges from 67% to 100% among adults and around 59% among children, with the horrifying statistic that the majority of deaths occur within a harrowing 24 hours of symptom onset.
24 hours.
It truly is a silent killer that demands immediate suspicion and action based on sometimes subtle initial signs.
Okay, let's really unpack the virulence now, the pathogenesis. Understanding how these infections cause such utter devastation is key to diagnosis and treatment. How does it start in traumatic gangrene?
Right. In traumatic gas gangrene, the process begins with the organisms being introduced directly into devitalized tissue that's damaged, lost its blood supply, and become anaerobic.
The perfect environment.
Exactly. For the strict anaerobes like C. perfringens and C. nophii, the initial trauma must be sufficient to interrupt the blood supply and thereby establish that optimal, oxygen-free environment required for their aggressive growth. The conditions are literally ripe for them to germinate from spores and explode in numbers.
But not for the aerotolerant ones.
These strict anaerobic conditions, however, are not strictly required for the more aerotolerant species, like C. septicum and C. tertium. As we discussed, these cunning bacteria can seed normal, healthy tissues hematogenously from gastrointestinal lesions, highlighting their more insidious nature.
Once they start growing.
Once introduced into an appropriate niche, whether it's damaged tissue or seeded normal tissue, the organisms rapidly proliferate locally. And most importantly, they begin to elaborate their incredibly potent exotoxins. These toxins are the true architects of the devastating disease we see. They are the weapons.
Let's zero in on the C. perfringens toxins, as it's the most common cause. The sources mentioned alpha and theta toxins being key, a symphony of destruction.
That's a good way to put it. Alpha toxin, theta toxin are highlighted as the major extracellular toxins primarily implicated in C. perfringens gas gangrene. They don't necessarily act alone, they often work in a coordinated, synergistic fashion to overwhelm the host.
Tell us about alpha toxin first. What does it do?
Okay, alpha toxin, this is considered the primary destroyer. It's classified as a lethal hemolysin, meaning it directly lyses or bursts red blood cells. But critically, its main destructive power comes from its enzymatic activities. It possesses both phospholipase C and sphingomyelinase activities.
Enzymes that break down lipids.
Exactly. Key components of cell membranes, phospholipids, and sphingomyelin. It essentially dissolves the membranes of host cells, muscle cells, blood cells, endothelial cells, lining blood vessels from the inside out, causing massive tissue damage and necrosis.
How do we know it's the main virulence factor?
Its status as the major virulence factor of C. perfringens is unequivocally established through several lines of evidence. Immunization of experimental animals, like mice, with just its C-terminal domain, a specific part of the toxin molecule provides significant protection against lethal challenge with live bacteria.
So targeting just that part works.
Yes. And even more convincingly, genetically engineered mutant strains of C. perfringens that are specifically deficient in producing any toxin they can't make it are found to be not lethal in animal models of gas gangrene. This really cements its role as the primary driver of lethality.
Any progress on treatments targeting it?
Yes, on a promising therapeutic front, the source mentions the recent development of a human single-chain recombinant antibody specifically designed to bind and neutralize toxin. This antibody has shown significant preventive and therapeutic efficacy in mouse models.
So a potential future treatment for humans.
It's a major step towards potentially having targeted antitoxin therapies for human patients, which could be a game changer alongside surgery and antibiotics.
How exactly does alpha toxin cause the specific features? We see the pain, the rapid spread, the lack of immune cells.
Great question. Experimental models have shown that the excruciating pain, the terrifyingly rapid spread of the infection along tissue planes, the marked tissue destruction, and that striking absence of neutrophils in the core of the lesion. These are largely attributable to a toxin-induced occlusion of small blood vessels.
Blocking blood flow. How?
What happens is fascinating and rapid. Within minutes of exposure, alpha toxin causes heterotypic aggregates of platelets and neutrophils to form within the microvasculature. Imagine the body's own tiny blood-clotting cells, platelets, and its key immune cells, neutrophils, clumping together inappropriately and physically, blocking the small blood vessels supplying the tissue. Figure 154-3 in the sources illustrates this beautifully.
Why do they clump together like that?
This aggregation appears to be largely mediated by toxin's ability to directly activate a key adhesion molecule on the surface of platelets, called glycoprotein IVA, GPIIA. It essentially flips a switch, telling platelets to become sticky and grab onto neutrophils and each other.
That has treatment implications, doesn't it?
Absolutely profound implications.
Yeah.
The idea posed is that existing platelet glycoprotein inhibitors, drugs like eptifibatide or abciximab, which are already used clinically to prevent platelet aggregation in heart attacks and strokes, might actually be therapeutic in gas gangrene for maintaining tissue blood flow and preventing this microvascular occlusion. It's a direct link from understanding the pathogenesis to identifying potential repurposable drugs.
Fascinating. Okay, what about the other major player, theta toxin, perfringens O?
Right, theta toxin, also known as perfringens O or PFO. This belongs to a different family of toxins called the thiol-activated cytolysins, specifically the cholesterol-dependent cytolysins, CDCs.
Are there other toxins like this?
Yes. Other notorious members of this family include streptolysin O, SLO, from group A Streptococcus, the bug that causes strep throat and rheumatic fever, and pneumocystin, PLY, from Streptococcus pneumonia, the common cause of bacterial pneumonia. So it's a well-known family of pore-forming toxins.
How do they work? They punch holes in cells.
Essentially, yes. These cytolysins function by binding as oligomers, groups of toxin molecules assembling together to cholesterol, which is abundant in host cell membranes. Once bound, they undergo a conformational change and insert themselves into the membrane, forming large, ring-like pores. At high concentrations, these pores lead to rapid cell lysis, the cell contents leak out, and the cell bursts.
But you mentioned sublytic effects earlier.
Yes, and this is crucial, maybe even more insidious. Even at sublytic concentrations too low to cause outright cell lysis or bursting theta toxin can still have significant damaging effects. It can hyperactivate immune cells like phagocytes and also vascular endothelial cells, the cells lining blood vessels.
Hyperactivate them how? What does that do?
By causing subtle membrane perturbations or signaling cascades, it can trigger these cells to release inflammatory mediators, increase vascular permeability, leading to edema, and contribute to the overall inflammatory storm and tissue damage, even without directly killing the cells. It's a more subtle form of attack.
Any other inflammatory roles?
Yes, theta toxin-mediated activation of the macrophage inflammasome has also been reported. The inflammasome is a critical part of the innate immune system, an internal alarm system within immune cells like macrophages. When triggered by PFO, it leads to the production and release of potent pro-inflammatory cytokines like interleukin-1, IL-1. This shows its deep role in driving powerful inflammatory responses that can paradoxically worsen the disease outcome.
So alpha toxin blocks blood vessels and directly kills cells, while theta toxin pokes holes and ramps up inflammation. How do they cause the systemic collapse seen late in the disease?
The systemic collapse, particularly the cardiovascular failure and end organ damage that tragically occurs late in the course of C. perfringens gas gangrene, is largely attributable to both the direct and indirect effects of both IL toxins working in deadly concert. They hit the system from multiple angles.
How does theta toxin affect the cardiovascular system?
In experimental models, A toxin, when infused systemically, causes markedly reduced systemic vascular resistance, meaning blood vessels widely relax or dilate, but interestingly, an increased cardiac output initially. This phenomenon is often referred to as warm shock.
Why does that happen?
It's probably mediated by the toxin inducing the release of endogenous mediators from host cells, things like prostacyclin and platelet activating factor. PAF. These are potent chemicals produced by the body that cause widespread vasodilation. It's a response very similar to what's observed in severe gram-negative sepsis, septic shock.
And alpha toxin's effect is different.
Yes, in sharp contrast to theta toxin's effect, alpha toxin appears to directly suppress myocardial contractility. It directly weakens the heart muscle's ability to pump blood effectively.
So it's cardiotoxic.
Directly cardiotoxic, yes. This leads to profound hypotension, dangerously low blood pressure, due to a sudden, drastic reduction in cardiac output. So you have theta toxin causing vasodilation and alpha toxin weakening the heart pump, a deadly combination leading to circulatory collapse.
A double whammy on the cardiovascular system.
Exactly. This highlights the distinct but powerfully synergistic roles of these two major toxins in driving the systemic deterioration and ultimately multi-organ failure.
Are other body chemicals involved? Cytokines.
While we understand a lot about alpha and theta toxin, the precise roles of other endogenous mediators in this complex process, things like various pro-inflammatory cytokines such as tumor necrosis factor, TNF, interleukin-1, IL-1 and interleukin-6, IL-6, or other vasodilators like bradykinin, have not yet been fully elucidated in the context of gas gangrene specifically. This suggests there are still critical areas of ongoing research and discovery needed to fully map the complex interplay between the toxins and the host response.
Okay, that's C. perfringens. What about the toxins of C. septicum, the other major player in spontaneous gangrene?
Right, C. septicum. It produces three main toxins that are considered important: an alpha toxin, which is lethal, hemolytic, and necrotizing, a beta toxin, and a gamma toxin.
Is its alpha toxin the same as the C. perfringens one?
No, and this is a key biochemical distinction. The alpha toxin of C. septicum does not possess phospholipase activity, unlike the potent phospholipase C activity of C. perfringens alpha toxin. So it works differently, likely through different mechanisms, although it's still damaging.
Do we know how these three toxins contribute?
The precise mechanisms of how these three C. septicum toxins, alpha, beta, and gamma, interact and contribute to the disease are still being actively elucidated. However, it is highly likely that each of these toxins contributes uniquely and perhaps synergistically to the devastating effects seen in C. septicum gas gangrene, making it a distinct and equally dangerous threat, possibly explaining its ability to spread in normal tissue due to its different toxin profile combined with its aerotolerance.
Okay, we understand the enemy's weapons now. Let's talk about fighting back. The critical aspects of treatment and prognosis for gas gangrene. What's the absolute first step if you suspect it?
Rapid and accurate diagnosis is paramount. Patients with suspected gas gangrene, whether it's the dramatic traumatic type or the more insidious spontaneous form, must undergo prompt surgical inspection of the infected site.
No waiting around.
Absolutely not. This isn't a condition for a wait-and-see approach or just starting antibiotics alone. Surgery is key.
What about lab tests? Gram stain?
Direct examination of a gram stain smear from the involved tissues or wound drainage is of major importance. It's rapid and can offer immediate, crucial diagnostic clues right at the bedside or in the operating room.
And what do you see under the microscope in tissue?
The characteristic histologic findings in clostridial gas gangrene, when a biopsy is examined, are quite distinct. You see widespread tissue destruction, muscle fibers looking necrotic and fragmented, but also that paradoxical finding, a striking paucity or lack of leukocytes, white blood cells, right in the most heavily infected tissues.
Even though it's a severe infection.
Exactly. This is found in conjunction with an accumulation of leukocytes, seemingly trapped within adjacent blood vessels, as illustrated in figure 154-4 in the source material. This unusual absence of immune cells at the core infection site, coupled with their buildup nearby, along with the presence of those large gram-positive rods, sometimes with spores visible, are crucial diagnostic features.
And that lack of immune cells is due to the toxins.
It's believed to be directly due to the potent effects of the bacterial toxins, like alpha toxin, which either kill the arriving leukocytes or prevent them from migrating out of the blood vessels effectively to reach the infection site.
What about imaging? X-rays, CT?
Imaging studies, specifically CT and MRI, are invaluable. They are excellent for determining the full extent of the infection and, critically, detecting the presence of gas within the soft tissues, which is a hallmark sign. They can help clinicians understand whether the infection is localized or, more dangerously, aggressively spreading along fascial planes, the connective tissue layers that surround muscles. This helps guide the extent of surgical debridement needed.
Can you diagnose it with just a needle aspiration?
While needle aspiration or a punch biopsy might provide an etiologic diagnosis confirming the presence of Clostridia in maybe about 20% of cases, it's critical to remember that these less invasive techniques should not replace immediate surgical exploration, gram staining of tissue samples, and thorough histopathologic examination of surgically removed tissue. You need to see the extent of the damage directly.
And that tip for spontaneous gangrene again.
Yes, a crucial tip. When spontaneous gas gangrene is suspected, blood should be cultured immediately, ideally before starting antibiotics if possible without delaying treatment. This is because bacteraemia, the bacteria entering the bloodstream, often precedes the appearance of visible cutaneous manifestations by several hours. Catching it in the blood early can be a life-saving diagnostic step.
Okay, diagnosis is rapid, suspicion is high. What's the absolute cornerstone of treatment?
Surgical management.
Yeah.
Without a doubt. For patients with any clinical or radiographic evidence of clostridial gas gangrene, thorough emergent surgical debridement, the aggressive removal of all dead, damaged, or infected tissue is of extreme and immediate importance. It is the single most critical intervention for survival.
How aggressive does the surgery need to be?
The surgical principle here is clear and non-negotiable. All devitalized tissue must be widely resected back to healthy, bleeding, viable muscle and skin. The goal is to remove every single bit of tissue that provides the anaerobic conditions allowing these organisms to continue proliferating and producing toxins. Sometimes this means multiple surgeries are required over subsequent days.
And should the wound be closed right away?
No. Critically, closure of traumatic wounds associated with significant contamination or tissue damage or compound fractures should always be delayed for maybe 5 to 6 days or until it is absolutely certain that the sites are free of infection. Closing the wound prematurely can trap any remaining bacteria in an anaerobic environment and lead to recurrence or worsening infection. Delayed primary closure or secondary closure is the standard approach.
Okay, surgery first and foremost. What about antibiotics?
Antibiotics are essential, but they are adjunctive to surgery. Except for that tricky C. tertium infection we keep mentioning, the primary antibiotic regimen for both traumatic and spontaneous gas gangrene, as outlined in Table 154.1, consists of a powerful combination of high-dose penicillin and clindamycin, usually given intravenously for 10 to 14 days or until the infection is clearly resolved.
Why both penicillin and clindamycin?
Penicillin is traditionally recommended based on historical in vitro sensitivity data. Most Clostridia were very sensitive to it. However, clindamycin is specifically and strongly recommended, often considered superior in this context.
Why is clindamycin preferred, even with penicillin?
Its preference stems from superior efficacy demonstrated over penicillin alone in animal models of C. perfringens gas gangrene, and also supported by some clinical reports and strong mechanistic reasoning.
Even though human trials are lacking.
Correct. It's important to acknowledge that large-scale controlled clinical trials rigorously comparing the efficacy of penicillin versus clindamycin versus the combination in humans with gas gangrene, have not been performed largely due to the rarity and rapid progression of the disease, making such trials ethically and logistically very difficult. But the strong mechanistic rationale and the compelling animal data overwhelmingly support the inclusion, if not the primary role, of clindamycin.
What if someone is allergic to penicillin?
In penicillin-allergic patients, clindamycin may be used alone as a primary agent.
What are the proposed reasons for clindamycin being better here?
The proposed mechanisms for clindamycin superiority are compelling and speak directly to the toxin-mediated nature of Clostridial infections. Key advantages include: first, its ability to inhibit bacterial protein synthesis, which means it can directly suppress the production of those devastating bacterial exotoxins, something penicillin doesn't do as effectively.
Shutting down the weapons factory.
Exactly. Second, its effectiveness is thought to be less affected by the size of the bacterial load, the inoculum size, or the stage of bacterial growth, unlike penicillin, which works best on rapidly dividing cells. And third, there's evidence suggesting clindamycin can modulate the host's inflammatory response, potentially lessening the damaging cytokine storm. These are profound advantages in fighting a toxin-driven disease.
But we talked about resistance earlier. Is that a major issue for these drugs?
It is a growing concern that absolutely needs to be considered. While C. perfringens historically has been largely susceptible to first-line antibiotics like penicillin and clindamycin, antibiotic resistance has indeed been increasingly reported globally.
Where has resistance been seen?
We've seen sporadic case reports of clindamycin-resistant C. perfringens causing infections like cellulitis or abscesses. More concerningly, larger surveillance studies from places like Canada and Taiwan have shown increasing rates of resistance to clindamycin among bloodstream isolates of Clostridium species over time.
Any specific numbers?
Yes, that significant 2014 Canadian study by Marchand Austin et al. Looked at over 1,400 anaerobic isolates. Among the 68 C. perfringens isolates, all were susceptible to penicillin, which is reassuring, but a notable 3.8% were resistant to clindamycin. And with the other Clostridium species they tested, nearly 300 isolates, the resistance rates were worryingly higher. 14.2% were penicillin resistant, and a substantial 21.6% were clindamycin resistant.
Wow, over 1 in 5 resistant to clindamycin for non-perfringens species.
Exactly. And more recent studies echo this concern. A study from Iran reported 21.2% of C. perfringens isolates resistant to penicillin. A 2019 study from Hungary, specifically looking at isolates from gas gangrene cases, found resistance rates in C. perfringens of 2.6% for penicillin and 3.8% for clindamycin. But for non-perfringens Clostridia causing gas gangrene in that study, resistance was higher, 6.8% for penicillin and 8.5% for clindamycin.
So resistance is definitely out there and seems higher in non-perfringens species. What's the takeaway for doctors?
The critical takeaway for clinical practice is that these findings, though variable by region and species, highlight the crucial and ever-growing importance of good anaerobic microbiology, including susceptibility testing whenever possible, especially for severe infections or treatment failures. This testing provides vital, up-to-date, potentially localized information, which is absolutely essential for making optimal clinical management decisions, especially given the rapid progression and high mortality of these infections. You can't just assume the standard drugs will always work anymore.
Okay, and reminding us about C. tertium treatment.
Right, always remember the exception. C. tertium is uniquely resistant to penicillin, cephalosporins, and clindamycin. Therefore, the appropriate antibiotic therapy for suspected or confirmed C. tertium infection is typically vancomycin, usually 1 gram intravenously every 12 hours, adjusted for renal function, or possibly metronidazole, 500 milligrams intravenously every 8 hours, although susceptibility testing is still ideal if available.
What about just finding Clostridia in the blood without full-blown gangrene? Clostridial sepsis.
For other Clostridial sepsis syndromes, Table 154.1 generally recommends that penicillin plus clindamycin combination again. Example, penicillin G 3 to 4 million units AV every 4 to 6 hours, plus clindamycin 600-900 milligrams IV every 6 to 8 hours. For penicillin allergy, alternatives include clindamycin alone, or possibly metronidazole or vancomycin, depending on the suspected species and local resistance patterns. However, it's also noted that finding Clostridia in a single blood culture without clear signs of systemic toxicity or an infection source might represent transient bacteraemia, possibly from the gut, which could be clinically insignificant and might not require aggressive or prolonged treatment. clinical correlation is key.
And if it's just wound contamination, no infection signs.
For simple wound contamination where Clostridia are cultured from a wound surface but there are no clinical signs of infection, like spreading redness, pus, systemic symptoms, antibiotic treatment is generally not required. Treatment should always be based on clinical signs and symptoms, not solely on bacteriologic findings from a surface culture. Good wound care is paramount.
Now, let's address the controversial topic of adjunctive hyperbaric oxygen HBO therapy. Sending patients into a high-pressure oxygen chamber. Does it help gas gangrene?
The value of adding HBO to the treatment regimen for gas gangrene remains a subject of ongoing debate and significant controversy among medical professionals. There isn't universal agreement.
What does the science say? Does oxygen kill these bacteria?
Basic science insights offer a mixed picture. High-pressure oxygen can inhibit the growth of the relatively oxygen-sensitive C. perfringens in laboratory settings. But importantly, it's thought to have little or no inhibitory effect on the growth of the more aerotolerant C. septicum. Furthermore, in vitro studies, experiments in test tubes have shown that the presence of blood and macerated damaged muscle tissue can actually inhibit HBO's bactericidal potential, suggesting its effectiveness might be limited in the complex debris-filled environment of a real wound.
What about animal studies?
Numerous animal studies investigating HBO for experimental gas gangrene have generally shown very little efficacy of HBO when used alone. In contrast, antibiotics alone, especially those that inhibit bacterial protein synthesis like clindamycin, confer marked and significant survival benefits in these models.
Does adding HBO to antibiotics and surgery help at all?
The evidence suggests that the addition of HBO to the standard therapeutic regimen, surgery plus antibiotics, does provide some additional benefit in some studies. But crucially, this benefit seems apparent only if aggressive surgical debridement and timely antibiotic administration precede the HBO treatment.
So surgery and antibiotics absolutely come first.
Unquestionably. Therefore, the critical takeaway for clinicians, emphasized by the source material, is that HBO therapy should absolutely not delay emergent surgery and the timely administration of appropriate antibiotics, particularly those that inhibit toxin production like clindamycin. These are the primary, proven life-saving interventions. Delaying them for HBO could be fatal.
Are there logistical issues too?
Yes, logistical considerations are significant. While some specialized trauma centers or wound care centers with hyperbaric oxygen units may have particular expertise in managing these aggressive infections, the potential benefits of HBO must be carefully weighed against the risks and time delays involved in transferring a critically ill patient to such a facility versus proceeding immediately with life-saving surgery and antibiotics at the presenting hospital. Time is tissue, and time is life in gas gangrene.
So a potential adjunct, but definitely not a primary treatment, and never at the cost of delaying surgery or antibiotics.
That sums up the current perspective well.
Given all this, what does the prognosis look like for someone who develops gas gangrene? What factors influence their outcome?
Several factors critically influence the outcome. The site of infection is a major one. The prognosis is generally more favorable when the infection involves an extremity, an arm, or a leg, compared to infections involving the trunk, like the abdominal wall or chest wall or visceral organs.
Why is location so important?
Simply because thorough surgical debridement of infections on the trunk or involving internal organs is considerably more difficult and often less complete than amputating or widely debriding a limb. It's harder to get clear margins and remove all the affected tissue.
When are the outcomes worst?
Gas gangrene is most likely to progress rapidly to systemic shock and death in patients who have associated bacteraemia. The bacteria have spread systemically, and especially those who develop intravascular hemolysis, which is the destruction of red blood cells within the blood vessels, caused by toxins like alpha toxin. Mortality rates are highest for patients who are already in shock at the time of diagnosis and initiation of treatment.
What about spontaneous versus traumatic?
Mortality rates are also relatively high among patients with spontaneous gas gangrene, particularly, as we noted, for cases due to C. septicum, likely due to delays in diagnosis, and the frequent association with severe underlying conditions like cancer or neutropenia.
And even if patients survive the initial infection?
Even for survivors, the journey is often incredibly long and arduous. They may undergo multiple debridement surgeries, require complex reconstructions, potentially lose limbs, and face long periods of hospitalization and intensive rehabilitation. It highlights the profound and lasting physical and psychological impact of these severe infections.
Given how devastating it is, prevention is obviously key. What are the main preventative measures?
Absolutely. Prevention starts with meticulous wound care. Initial aggressive surgical debridement of devitalized tissue is crucial to reduce the risk of gas gangrene developing in heavily contaminated deep wounds, especially those from trauma. You have to remove the potential anaerobic breeding ground right away.
What practices should be avoided?
It's also critical to highlight specific interventions to absolutely avoid. Prolonged application of tourniquets should be avoided as much as possible, as this can create ischemia and subsequent anaerobic conditions distal to the tourniquet. And importantly, as we mentioned regarding surgery, premature surgical closure of heavily contaminated traumatic wounds should be avoided. Patients with compound fractures, for instance, are at significant risk for gas gangrene if their wound is closed surgically early on, as this can inadvertently create the perfect oxygen-free environment for any contaminating Clostridia to thrive. Open wound management initially is often safer.
Any progress on vaccines for prevention?
On the research front, as we touched upon, vaccination against alpha toxin has proven protective in experimental animal models of C. perfringens gas gangrene, demonstrating proof of concept. However, this has not yet been investigated in human clinical trials.
And the hyperimmune globulin?
And as mentioned previously in the epidemiology section, the development of a potent hyperimmune globulin containing antibodies against key Clostridial toxins represents a significant potential advance for prophylaxis, giving it preventatively to high-risk trauma victims or for use therapeutically to attenuate the spread of established infection. It remains a promising area for future development.
While gas gangrene is certainly the most infamous, let's not forget some of the other notorious Clostridial infections that pose significant, often stealthy threats. First, you mentioned Clostridium sordeli infection earlier. This sounded particularly nasty and hard to diagnose.
Yes, C. sordeli infection can truly be a silent, stealthy killer. Historically and primarily, it's been associated with gynecologic procedures, childbirth, both vaginal delivery and C-section, or abortion, whether spontaneous, elective, surgical, or medically induced, for example, using mifepristone ibuprostol.
And it progresses quickly.
Once established, the infection proceeds with terrifying rapidity, often leading to a distinct form of toxic shock syndrome, TSS, and tragically frequently death within just a few days.
What makes its TSS unique? You mentioned specific features.
Yes, the systemic manifestations of C. sordeli CSS are quite distinct from typical staphylococcal or streptococcal TSS. It's characterized by massive generalized edema, swelling from leaky capillaries, large effusions, fluid buildup in body cavities like the chest or abdomen, profound leukocytosis.
Extremely high white blood cell count.
Extremely high, often exceeding 50,000 or even reaching 100,000 to 200,000 cells per microliter, with a significant left shift, indicating a flood of immature white cells. This is accompanied by marked hemoconcentration, where the blood becomes unnaturally thick due to fluid leaking out of the vasculature, with hematocrit levels sometimes rising to an incredibly high 75% to 80%.
Wow, that's almost sludge.
It practically is. This whole constellation is then followed by the rapid onset of refractory hypotension shock that doesn't respond well to fluids or vasopressors and multi-organ failure. The lab findings of the massive white counts, the high hematocrit, are particularly striking and highly characteristic, acting as a critical red flag if recognized.
But the real kicker is the lack of typical infection signs.
And here's the truly insidious part that makes early diagnosis incredibly difficult and often fatally delayed. Significant pain may not be a prominent feature, especially early on, and crucially, fever is typically absent or minimal.
No fever and a deadly toxic shock. That's counterintuitive.
Highly counterintuitive. This is a critical departure from many other severe infections, which almost universally present with pain and fever as early warning signs. This lack of fever in C. sordeli TSS is perplexing and contributes significantly to diagnostic delays.
What are the fatality rates like?
They are distressingly high. In one case series review cited in the sources, the case fatality rate was a staggering 100% in cases associated with normal childbirth, 100% with medically induced abortion, and still high even with spontaneous abortion. Of infections not related to gynecologic procedures or childbirth, about 22% occurred in injection drug users, with a 50% mortality rate in that specific group. Other infections followed trauma or surgery, often in otherwise healthy persons with a 53% mortality rate.
So extremely deadly across the board.
Overall, the mortality rate in that large series was a staggering 69%. Nearly 7 out of 10 patients died. And notably, 85% of those who succumbed died within a harrowing 2 to 6 days after the onset of infection, or following the inciting procedure, underscoring the terrifying speed of this disease.
Is C. sordelia the only one causing this kind of picture?
Briefly, it's worth mentioning that a very similar syndrome, Rapidly fatal spontaneous necrotizing endometritis, infection of the uterine lining with toxic shock, extreme leukocytosis, leukemoid reaction, and capillary leak has also been described with Clostridium bifrumens, showing that other Clostridia can occasionally mimic this aggressive pattern.
Why is C. sordelii so hard to diagnose early?
The challenges in early diagnosis are manifold. First, the overall prevalence of these infections is low, so they simply aren't top of mind for many clinicians when a patient presents with vague symptoms.
And the symptoms themselves.
Second, the initial symptoms are often nonspecific and frankly misleading, maybe some nausea, vomiting, diarrhea, weakness, flu-like malaise, often resembling more common self-limited viral syndromes.
And the lack of fever is key.
Absolutely. Given these vague initial symptoms and, crucially, the perplexing absence of fever, physicians often don't initially feel the urgency to aggressively pursue additional diagnostic tests or consider a severe bacterial infection, which leads to precious diagnostic delays.
Especially if there's no obvious wound.
Exactly. The absence of obvious local evidence of infection, like a visibly infected wound or abscess, and the lack of fever, make early diagnosis particularly problematic in patients who develop these deep-seated infections following childbirth, therapeutic abortion, gastrointestinal surgery, or even seemingly minor trauma.
What do doctors think it is initially?
These patients, presenting with shock, edema, maybe shortness of breath from effusions, but no fever, are frequently evaluated for other conditions that can cause shock without fever, things like pulmonary embolism, internal gastrointestinal bleeding, sometimes pyelonephritis or cholecystitis if there's abdominal discomfort. This diagnostic detour further leads to critical delays, while the Clostridial infection and toxin production rage internally.
And the consequences of delay are severe.
Unfortunately, such delays in diagnosis significantly increase the risk of death. As in most rapidly progressive necrotizing soft tissue infections, by the time clear local signs and symptoms become apparent, if they ever do, and the correct diagnosis is suspected, patients are often already profoundly hypotensive, with irreversible organ dysfunction well established.
You mentioned injection drug users might be diagnosed earlier.
Yes, the one exception where the infection might be suspected somewhat earlier is in injection drug users who present with more typical local signs, like swelling, pain, and redness at their injection sites. This earlier recognition of a localized infection likely contributes to the somewhat lower, though still high, mortality rates observed in this specific group, as timely surgical and antibiotic intervention becomes more feasible.
So, summing up the clinical suspicion for C. sordelii, what's the key takeaway?
The absolute key takeaway for clinical suspicion is this. Physicians should maintain a high index of suspicion and strongly suspect C. sordelii infection, or potentially C. bifrumens. In a febrile, remember, no fever patients who present within roughly 2 to 7 days after an injury, surgery, drug injection, childbirth, or abortion, AMD, who report this seemingly nonspecific constellation of symptoms including pain, which might be abdominal, pelvic, or elsewhere, nausea, vomiting, and possibly diarrhea, often accompanied by weakness or malaise. That specific cluster of symptoms without fever, especially in the context of a recent potential exposure event, should raise an immediate life-saving red flag.
What about treatment? If it's diagnosed so late, is treatment effective?
When it comes to treatment limitations for C. sordelii, there is tragically little solid information regarding the most appropriate treatment, primarily due to the rapid and fulminant course of the disease. As we said, the interval between the onset of symptoms and death is often so short that there's little to no time to even initiate, let alone evaluate the effectiveness of empirical antimicrobial therapy.
And testing takes time too.
Compounding this, anaerobic cultures of blood and any wound aspirates are inherently time-consuming to process, and many hospital laboratories do not routinely perform antimicrobial sensitivity testing on anaerobes like C. sordelii, further hindering targeted treatment even if the organism is identified.
Is it usually susceptible to standard antibiotics?
Older, limited antibiotic susceptibility data suggests that C. sordelii, like most Clostridia, is generally susceptible in vitro to beta-lactam antibiotics, like penicillin, ampicillin, clindamycin, tetracycline, and chloramphenicol. It's typically resistant to aminoglycosides and sulfonamides. However, in vitro susceptibility doesn't always translate to clinical success, especially with toxin-mediated diseases.
Could toxin suppression help, like with clindamycin?
That's the hope. Given the likely primary role of toxins in C. sordelii pathogenesis, antibiotics that are known to suppress bacterial toxin synthesis, with clindamycin being the prime example, may possibly prove useful as therapeutic adjuncts, similar to their proposed benefit in gas gangrene and streptococcal TSS. However, definitive clinical data proving this benefit specifically for C. sordelii TSS is lacking. early aggressive supportive care and potentially surgery to remove any infected source, like retained products of conception, are likely critical.
Okay. Lastly, let's briefly touch on other significant clostridial skin and soft tissue infections mentioned in the sources, beyond gas gangrene and C. sordelii. What else is there?
One condition mentioned is crepitant cellulitis, sometimes called anaerobic cellulitis. This primarily occurs in diabetic patients, often affecting the lower extremities.
How is it different from gas gangrene?
It characteristically involves the subcutaneous tissues just under the skin, or sometimes retroperitoneal tissues behind the abdominal lining. Gas production, crepitus, is common, giving it its name. But notably, it typically does not involve the underlying muscle, myonecrosis, or the deep fascia, distinguishing it pathologically from true gas gangrene or necrotizing fasciitis.
Is it less serious then?
While it might be less invasive initially, it should not be underestimated. Despite the lack of deep tissue invasion, it can still progress rapidly and lead to fulminant systemic disease and sepsis, if not treated promptly with antibiotics and sometimes surgical drainage or debridement.
Any other specific species mentioned?
Yes, cases of C. histolyticum infection causing cellulitis, abscess formation, or even endocarditis, an infection of the heart lining, have also been documented, again, particularly in injection drug users. C. histolyticum is known for producing potent collagenases and proteases, contributing to tissue breakdown.
Infections inside the eye.
Endophthalmitis, a severe infection inside the eye that can lead to blindness, has been described due to direct inoculation of C. sordelii or C. perfringens, usually after penetrating trauma to the eye.
And C. ramosum. You mentioned it in mixed infections.
Yes, C. ramosum is highlighted as another species that is frequently isolated from various clinical specimens, including blood cultures, intra-abdominal infections, and soft tissue infections. It demonstrates its diverse pathogenic potential and its widespread clinical relevance, often as part of those polymicrobial infections we discussed earlier. Hashtag tag outro Atra.
And there you have it. Wow. That was truly a deep dive into the fascinating yet undeniably formidable world of the Clostridium genus. We've journeyed from their remarkable diversity and that potent toxin arsenal, really understanding the biochemical wizardry that makes them so dangerous,
The sheer variety of weapons is incredible.
to the devastating clinical syndromes they cause, especially the truly life-threatening gas gangrene, both traumatic and spontaneous, and those insidious, hard-to-catch C. sordelii infections.
Infections that demand constant vigilance.
The key message from this deep dive seems crystal clear. Understanding not just what these infections are, but the intricate why and how behind their mechanisms, their epidemiology, their variable presentations, and their frighteningly rapid progression. Understanding all that is absolutely crucial for recognizing them early, diagnosing them accurately, and ultimately combating them effectively.
Absolutely. It's about knowing the enemy inside and out, understanding its playbook, the toxins, the need for anaerobiosis, or the lack thereof, the subtle clinical clues, so you're less likely to be caught off guard when facing these critical situations.
Thinking like that elite professional we mentioned at the start.
Indeed. And as you, our listeners, reflect on all this information, maybe think about how even the smallest details we discussed, like a specific toxin's enzymatic activity, its effect on blood vessels, or a subtle change in a bacterium's oxygen tolerance, or the significance of finding C. septicum in the blood, how these seemingly small biological details can have such profound and often catastrophic implications for human health.
It really highlights the complexity.
It does. I mean, consider the constant arms race playing out between these microscopic pathogens and human medicine. There's always more to learn, more toxins to understand, more resistance mechanisms emerging, more diagnostic challenges. So perhaps a final thought to leave you with. What stands out to you as the most surprising or unsettling aspect of Clostridium's silent yet deadly arsenal that we explored today? And perhaps more importantly, what new questions does this deep dive spark in your mind about the delicate balance of our own microbiomes, the power of bacterial toxins, or the critical, often razor-thin importance of rapid diagnosis and decisive medical intervention in a world where microscopic threats can escalate with such devastating speed? Something to ponder.